Hi, Mr. Az here. Welcome to our revision session on inheritance and evolution. Today we're going to be looking at the focus 5 of DNA structure, meiosis, the use of Punnett squares, genetic engineering and classification. I will say that this is not the only things you need to be revising. There are selected topics that we consider to be important, that we know come up a lot, and we also know that can often lead to misconceptions. So please, this is not the sole revision you should be doing on this topic. Please use other resources supplied by your teacher. So starting off, we're looking at DNA structure. Here we can see a normal eukaryotic cell. We've got the nucleus, and within the nucleus we have chromosomes. Here I've zoomed in on a chromosome. It's a typical structure. One chromosome is simply one strand of DNA supercoiled up and stuck together with proteins. Here we can see the strand of DNA coming out from the chromosome. One section of this DNA that codes for a particular feature or characteristic or protein we call a gene. We see that DNA is a double helix, which means there are two strands of DNA and they intertwine around each other in a helix form. DNA is considered to be a polymer. It's a repeating unit of monomers, and this is a single unit here which makes up the DNA. Now, you do not need to know what a base is. You do not need to know what a phosphate group is. I think you kind of understand what a sugar is. Now, as we move to A level, the complexity of this increases a bit, but for now, you just simply need to know that the phosphate and the sugar part of this monomer make up the backbone of the DNA, so that grey bit here. The base will be the section that joins the two strands of DNA together. Here we can see how the polymer is formed. We have the sugar phosphate backbone, and we have the bases here joining to complementary bases across to join the two strands together. In DNA there are four types of base. There's adenine, thymine, guanine and cytosine. Fortunately, you don't need to know the names. What you do need to know are the letters. So A, T, C and G. Complementary base pairing is where the bases will always form a bond with the same bases. So thymine will always form a bond with adenine. Guanine will always form a bond with cytosine. Moving on now to meiosis. Meiosis is a form of cell division. Similarly to mitosis, it will form part of the cell cycle. The whole process or cell cycle is the increasing in size of a cell before it goes into cell division. So prior to cell division, the cell will increase its cell content, so things like mitochondria are duplicated, ribosomes are increased in number. We also see in this process the chromosomes duplicating. We see DNA replication and these chromosomes duplicating, so that when we move into the actual cell division phase up here in meiosis, we have a large enough cell to split into daughter cells. And the other key bit is that the chromosomes obviously have increased in number. They've duplicated and doubled in number. We know that there are 23 pairs of chromosomes in a human somatic cell or a human body cell. Each pair of chromosomes forms part of a homologous pair of chromosomes, so they code for the same features. They might have different alleles on each chromosome, but they code for the same features. Here we see a pair of homologous chromosomes and another pair of homologous chromosomes here. As I said, before we move into meiosis, before we move into cell division, the chromosomes have duplicated. So this chromosome has been duplicated to form two. So this chromosome that's been copied is exactly the same as the chromosome here. Likewise, this chromosome here has been copied to form an exact copy of this chromosome. You do not need to know the stages of meiosis and mitosis for that matter. You might have been taught prophase, metaphase, anaphase, and telophase, PMAT. You do not need to know them. However, if you would like to talk about them, you can in your exam. This stage here, we can see the chromosomes, the homologous chromosomes, being pulled to opposite sides of the cell. Okay, this phase we would call anaphase. The cell will split into two, and we are given two daughter cells at this point. Again, we're getting a second movement of the chromosomes to opposite poles of the cell. This process creates 4 new nucleuses. We can see here that each nucleus will have 23 chromosomes. Okay, so that is half the number of chromosomes that the cell had in the very beginning, the parent cell had in the very beginning. These cells will then split to create 4 distinct daughter cells. Here you can see the 4 daughter cells, and we've made 4 sperm cells here, gametes. The end product of meiosis in humans will be gamete production. So we've got 4 sperm cells here, each with 23 chromosomes inside the nucleus. It's nice to compare meiosis and mitosis. We use a table for this. We've got meiosis down this side, mitosis this side. In meiosis, we create 4 daughter cells. Mitosis, just the 2 daughter cells. Meiosis creates daughter cells that are different to each other, genetically different to each other, and they're also genetically different to the parent cell. This is obviously an advantage in gametes because sexual reproduction creates differences in offspring. Genetic makeup in mitosis is that each daughter cell is identical to each other, and it's also identical to its parent cell. The number of chromosomes in meiosis is that there's a half set, so 23 chromosomes, and in mitosis you have a full set of chromosomes, 46, identical to the parent cell. Meiosis is used, as I said, in the production for gametes in sexual reproduction. Mitosis, production of body cells, growth and repair, and it's also seen in asexual reproduction in bacteria. Okay, now we move on to the use of Punnett squares to look at inheritance. In this slide we can see some of the key terminology we need to talk about when we talk about inheritance. I suggest you pause the video at this point and read through the keywords. Okay, we know that each body cell in a human has 23 pairs of chromosome. This is a representation of those 23 pairs of chromosome. As I mentioned before, when we're talking about chromosomes, a pair we call a homologous pair of chromosomes. So that means that they code for the same characteristics. They might, though, code for different versions of that characteristic. We call this an allele. So each allele on each chromosome will code for the same characteristic, but perhaps a different version. We can see in this example we have 2 different alleles. This allele is dominant, this allele is recessive. Dominant means that if the allele is present, the characteristic will always be shown. If it is recessive, the characteristic will only be shown if both sets of alleles are recessive. So in this case, the expressed gene or the expressed characteristic will be the dominant characteristic. If this individual were to produce gametes through meiosis, so the process we've just discussed, the end result, the gametes, the daughter cells, would be either a big B or a little b. So given the cell that produced the gametes has a big B and a little b, each gamete produced would either be a big B or a little b. It's a 50-50% chance of producing a dominant allele and a recessive allele in the gametes. If this individual then were to reproduce with another individual who for this instance has a large B and a large B, so they are homozygote dominant, then you will get these gametes produced by this individual, and we'll get these gametes produced by this individual. We use Punnett squares to calculate the probability of the offspring's genotypes. This is a Punnett square, dead easy to draw, just draw a hashtag. This side we will place in the the alleles from individual 1, and up here we place in the alleles from individual 2. So remember, it is the probability of production of gametes here. It can only, this individual can only produce big B and a big B in the alleles of the gametes. This individual can produce a big B and a little b in its gametes. So we place those alleles into the Punnett square. We then do a cross, so we just fill in the gaps here. So if an offspring here, so these represent the genotypes of the offspring. If an offspring is created from this gamete and this gamete, we would see a big B genotype. Again, if we have an offspring here produced from these gametes, we have another big B. Down here, we've got a big B and a little b gamete producing a big B, little b. We call this heterozygote. And again, in this corner here, we've got another big B, little b. So the result is this: every single possible offspring from this cross will have the dominant allele. Therefore, 100% of offspring will have the dominant trait. Okay, so for this cross, we've crossed a heterozygous individual with another heterozygous individual. So having a dominant and a recessive allele up here, a dominant and recessive allele here. The cross is different now, so we have another individual down here, possible offspring down here with a little b. So this offspring is the only offspring that would have or exhibit the recessive trait. 75% of the offspring have the dominant genotype, the dominant phenotype, and we might also represent this in a different way. We might also say that it's a 3:1 ratio. So every cross that occurs, the chances of it being dominant is 3, the chance of it being recessive is 1, so a 3:1 ratio. If you fancy a challenge, this is a question. You might want to pause the video now and have a go at this. It's tricky, so if you're up for a challenge, have a go at this. Okay, so we're told that night blindness is caused by a dominant allele. If the individual here is clear, so it's a white circle or white square, we're told that they are normal. Awful term, but they are told they're normal, which means they do not have the condition. Therefore, they can only have one genotype. You may want to pause and have a think about this, but these have to be little n, both of these individuals. I can also assume that this individual X, has to get one of its alleles from this parent. Okay, there's no other way that it can get an allele apart from one from this parent and another from this parent. So this individual has to have a little n. It has to have a recessive gene here. The genotype of person X therefore has to be big N, little n. Okay, so the cross is person X marrying someone normal site, so that individual would be little n. If we do a quick Punnett square over here, we have individual X, big N, little n, and the person they're crossed with little n, we have a big N, little n, big N, little n, little n. Okay, so the answer to this is a 50-50 split. We can see here that those two individuals have a dominant allele, so they will have night blindness, and these two individuals here, possible individuals, offspring, have little n, so they are normal sighted. So the answer is 50%. Okay, moving on to genetic engineering. Okay, so genetic engineering is simply the changing of the genome of an organism to for a gain. Okay, and in this case we're looking at insulin, the production of insulin. So we might want to make insulin to give to people who are diabetic. The start is to take a cell from a human who produces insulin, so they do not have the faulty gene. Okay, we take the cell out of the individual, we look at the correct chromosome, and we isolate the correct gene. So we've got the insulin gene here. So how do we isolate the gene? Well, we use something called a restriction enzyme. This enzyme actually cuts DNA. So this enzyme will cut the DNA in a specific way and it will target the specific gene. So we've isolated and we've removed that gene. Okay, so we've now isolated the gene. What do we do with it? Well, we're going to place it inside a vector, inside something that carries the gene into an organism. Okay, and in this case we're looking at plasmids. So we remove the plasmids from a bacterium. Remember, plasmids from cell biology. The plasmids are removed from the bacterium, and again we use a restriction enzyme to cut the plasmids. So we're cutting the DNA so that we can insert the insulin gene. Okay, so now we can see the gene has been inserted into the plasmid. We're then going to place that plasmid back into the bacterium. So this bacterium now contains the plasmids containing the human insulin gene. So the bacterium will create insulin, produce insulin just by its own ribosomes. So the ribosomes, if you remember, produce proteins. They make proteins. They're the factories for making proteins. Bacterium have ribosomes as well, and the bacterium is tricked into producing human insulin. So here we can see the human insulin being created within the bacterium. Okay, so large-scale production. We use a fermenter, which is a device where you just shove a load of the bacterium and a load of the right conditions, so warm temperature, moisture, a food source, and the bacteria will grow and grow and grow, and they'll produce a whole load of human insulin. That human insulin is just tapped off and placed into vials, and then can be used for treating people with diabetes. So last of all, thanks for sticking with me, is classification. Classification is just a way of identifying and grouping organisms on this planet. So every organism on this planet should be classified into a particular group based on its characteristics. Here we're going to be classifying a lion. Okay, so the order as we move down through the classification system is kingdom, phylum, class, order, family, genus, species. You might want to think of a mnemonic for remembering this. I've heard some good ones along the way. The ruder the better. The top, each organism on the planet will fit into one of 5 kingdoms. The lion fits into the animal kingdom. Next is chordata, which is vertebrates. So the lions have a vertebrae. They are classified under this section. Next, it's a mammal. Mammalia, carnivore, family, Felidae. So this is the cat family. Genus, Panthera. And we have the species Panthera leo. As you move up towards the kingdom, the number of organisms within that group increases, but the similarities decrease, if that makes sense. I would also suggest that the key ones are the kingdom and the genus and species. So between, you don't get many questions on the groups between kingdom, genus and species. Okay, so focusing in on the genus and species bit, we can see this is called the binomial system. So here we've got a 2-word description of the species, the first being the genus, the second being the species name, so Panthera leo. This should be written in italics and it should also be underlined. binomial system, each species, so if you see a species, if you see a name that is underlined and in italics, is going to be the species name. There will always be two of them, and they should always be in italics. They may be underlined. If you get a question on perhaps naming the genus for this species, well, it's simply taking the first word, because the first word is the genus, and that will always be true. One final note on classification is that we have a new system. We have a time, if you think about the classification system, Carl Linnaeus, is around in the 18th century. It's been around for a long time, and his systems were based on visual characteristics. As we move through time, technology's changed, our understanding of organisms has changed, and we have a new system whereby we can classify into 3 domains. This is work done by a guy called Vos, and he looked more at the biochemistry and the genetics of organisms, and grouped them based on, I guess, some more refined information. So we have 3 domains. We have archaea, we have bacteria, and we have eukarya. So that kind of finishes the revision session. I hope you found it useful. Please, as I said before, this isn't everything you need to know from the topics. However, I think it's a good start. Good luck with your revision, and thanks for listening.